This is our video lab report number 7 on polymerase chain reaction, presented to you by group number 4, Massiel Martinez, Juan Osiel Sanchez, and Mauricio Sanchez. Introduction. For this PCR lab, the purpose of it is to gain an understanding of performing polymerase chain reaction and using gel electrophoresis for separating and analyzing DNA based on its size and charge. The materials and methods for this lab. In this lab, we used the PCR thermocycler, 25 microliter PCR tubes, the food samples suspected to have E. coli, the PCR master mix, which contains Taq DNA polymerase, the DNTPs, the buffer, and the magnesium chloride, the forward and reverse primers specific to the E. coli gene, the nuclease-free water, positive control DNA, negative control with no DNA template, the electrophoresis equipment and gel, DNA ladder marker, gel loading dye, and agarose gel. The procedure for this lab. We begin by labeling the PCR tubes according to the samples that were used to be tested. So food sample 1, food sample 2, positive control, and negative control. Then we prepared the PCR master mix by combining the following components: the 1 microliter of forward primer and 1 microliter of reverse primer to each PCR tube, then 1 microliter of DNA template to the corresponding PCR tubes, and 22 microliters of the PCR mix is going to go into the PCR tubes, and then we will vortex this to ensure that it's properly mixed, and then amplify the DNA by placing the PCR tubes into the PCR thermocycler. And the conditions that we're going to use are 94 degrees Celsius for 60 seconds for the initial denaturation, and then 94 degrees Celsius for 30 seconds, again for the denaturation, and then for annealing, 57 degrees Celsius for 30 seconds, for extension, 72 degrees Celsius for 30 seconds, and the number of cycles that we used is 30 cycles. And then for the final extension, we're going to use 72 degrees Celsius for 60 seconds. And the final steps in the procedure are the gel electrophoresis steps. You will begin by preparing a 1.5% agarose gel using TAE buffer and adding gel red or another DNA stain to the gel. You will load the PCR products into the wells of the agarose gel using the DNA ladder marker for size reference, and you're going to run the gel at 100 volts for about 30 to 40 minutes until the bands are sufficiently separated. And finally, you'll visualize the gel using the UV light and analyze the PCR results to determine the presence or absence of the E. coli gene in the food samples. And you'll compare the results of the positive and negative controls as well and determine the estimated size of the DNA fragment. Observations for the PCR. In our lab journal, some of the observations we wrote are written here. The dye from each well slowly moved across the gel. When we looked at it under the UV light, we saw the DNA samples highlighted, and then we also saw that the smaller sized DNA molecules moved faster, and the DNA ladder or the reference that we used showed that the size of the DNA was 0.300 kb.
So here are our results after performing a PCR on different samples that we contained. As you can see here under this picture, this is a PCR conducted after running through denaturation, annealing extension, and running through numerous cycles. Then after running that PCR, we then inserted that sample into a designated electrophoresis, which is performed here. So as we can see those very green fluorescent bands, beginning with the very left band, that is our positive control, that just means that this band fully contains the E. coli. Now, to the right of that, where there is no band, that was our negative control, and this just tells us that there is no E. coli present. And then those following three basically aligned bands are different unknown samples that we had, and we were testing if they all contained E. coli. As we can see, that they all correspondingly do contain E. coli as they match with the positive band on the very left. And to the right of that, we have our ladder, and that ladder is just basically to measure the number of kilobases. And so, just summing up everything, basically, when running a PCR on E. coli with the mentioned samples, the expected results would typically involve the amplification of DNA segments specific to that E. coli genome. The control positive to the very left sample is known to contain E. coli DNA, which should show those distinct bands on the gel corresponding to the amplified DNA segments. Then those three samples on the right, which were labeled as A, B, and C, we can see that those also contain E. coli, which exhibit the distinct bands on the gel similar to that of the control positive sample, indicating the presence of E. coli DNA. These bands are typically visualized with gel electrophoresis, where the DNA fragments are separated based on their size and charge, allowing for a clear distinction between the different samples. And based on our results, we concluded that our sample size is at 0.3 kilo bases. And just real quickly, those bands started at the very top. As we know, DNA is negatively charged, and as the electrophoresis is running, electric current is running from the negative charge all the way down to a positive charge. So after 30 minutes, our DNA sample traveled that far, which thus giving us that size of 0.3 kilo bases.
Conclusion. What we concluded by the end of the lab was that the DNA fragment size was 0.300 kb or kilobases, and that polymerase chain reaction is a very important technique that helps to amplify DNA sequences and replicate DNA to detect and analyze it. And then with gel electrophoresis, we can separate these DNA fragments based on their size and visualize and analyze them, and they are fundamental tools for scientific research and practical applications in various areas. And this is the end of our video lab report number 7 on PCR. Thank you for watching.